Swayam Prabha. Digital India. Educated India.
Hello everyone, welcome back to our sessions on Preventive Oncology course. We have been continuing with cancer detection method. So it's actually why we are going for the third session for this particular topic is because of the importance of cancer detection using precision oncology or using. So as we have been always, I've been always been mentioning, right, no more one size fits all. It is like each patient has to have the therapy and the diagnostic tailored to their tumor molecular signatures. So last two sessions we've really dealt with what are exactly cancer detection methods, what exactly are biomarkers, what exactly are the detection methods for different cancer types, for your breast, for your lung and all that. Now because of this importance, now it's like this is a very much an emerging and in the much needed field, this particular cancer detection or the diagnostic methods using molecular technologies. So instead of going further into the molecular technologies, I'm now confining to the tumor liquids biopsies in this particular session. So what do you mean by a tumor liquid biopsy? So basically we will look at what are the so this is a very much a preferred tool of sample collection for cancer therapies and cancer diagnostics now. So the whole diagnostic, the molecular signatures for a particular tumors are basically now traveling towards this particular sampling of tumor liquid biopsy. Basically as we go on towards the end of the session, you will completely understand what is exactly tumor liquid biopsy, why is it advantageous over your regular biopsy methods, and it is much more preferred because of its non-invasive nature. So before we further go on, so just a very quick brush up of how do you stratify the molecular of human cancers. So it's just not like a complete broad cancer types. You even have your for example your ER minus and your ER pairs, where you have your luminal A positive, luminal B, and then you have the BRCA. Then for your colorectal you have the your CIMPs, MMR, and very importantly the most heterogeneous of all, one of the most heterogeneous of all cancer types, your lung cancer, non-squamous cell lung carcinoma. See how many different stratifications is there. Just imagine for one patient, for one particular tumor, can we have drugs for all this particular driver mutations? Yes. So if you aid a clinician in exactly identifying what mutation the patient harbors, it really helps the clinician to choose a particular inhibitor, for example a TKI inhibitor if they are EGFR positive. So similarly for your prostate you have the different stratification, and for the melanomas you have the BRAF mutations. So for the gastric it's the HERT, and for mostly for your pancreatic you have the KRAS and all. So now usually it's like as we all know, cancer arises because of a consequence of mutations in genome of cancer cells. So comprehensive views of cancer genome also there is always reveal that there is always a lot of heterogeneity among all tumors, most of the tumor types, and it is very important, this molecular taxonomy of all this tumor, for example for your solid organ tumors, it has to be informed so as to aid clinical decisions and treatment and the level of sensitivity of conventional clinical trial approaches, they have to be evaluated. So this is very important. So this degree of fractionalization or your stratification, it may increase over the time as much as the knowledge of this particular molecular signatures of each cancer type is totally increasing. So there is like so there as much there are the phenotypic tails of multiple variants are long. So these cancers where dominant phenotypes are known to exist, the residual subtypes like here for example may be very complex or heterogeneous. So for example, so that is why they may be, for example here this may be a bit difficult to treat. So for example, so now what we saw here earlier slide, we just show how the classification of cancers according to the different organ specific. Now for example, you just reverse your X and Y here. So you have your biotype classification. So estrogen dependent is the breast, then you have the several subtypes, then ABL like kinase dependent. So these are the particular different types of cancers. So then you have your EGFR dependent, here you have your lung cancer and the colon cancer. Then if you have your HER2, you have your breast, gastric and the pancreatic or the PDAC. So again you say ALK inhibitor responsive. So you are stratifying, you are classifying according to the particular drug. So for you have your lung cancer and all. Then your homologous recombination defective, you have the breast, ovary and the pancreatic. Then for the BRAF mutant, you have the melanomas. So like that, so on. This is again your based on your biomarkers or your what we have discussed. So you have already classified cancer here. So this is this is a simple algorithm which is already recommended by the National Societies of Cancer. So for predictive BRCA testing in a particular tumor. So here patients with recurrent high grade serous ovarian tuberosity or tubal or primary peritoneal carcinoma, they are considered for an olaparib maintenance therapy here. So whereas for patients who have unknown BRCA status or patients who could have been tested negative for BRCA germline mutation, you know, so there where if they have BRCA mutations, there is like this is it is eligible for therapy. So patients who have previously been tested positive for germline BRCA mutations are eligible for therapy and they do not need further testing actually. So this is a algorithm for predictive BRCA. So similarly you have your algorithm for your non-squamous cell lung cancer molecular testing. So this is like in our institute, it's a very well established molecular testing. So we adapt here the qPCR. So and the NGS as well. So here what happens is like first after the patient, so how do they take the biopsy? So once the patient comes with a stage 4 or a stage, usually mostly patients present here and the later stages, stage 3 or 4. So the biopsy bit, an excision biopsy is taken and it is sent for molecular testing in the EGFR and the ALK-RETROS. So we do this particular panels here. So and here in the EGFR, if the patient is positive, they go for the TKI inhibitors. So that's how the targeted therapy, your TKIs are administered for EGFR positive. So whereas in the ALK also we have the qPCR, but there is an ALK IHC also, it's recommended here where you have the IHC positive and the ALK IHC and or even the FISH also they do that. So suppose if negative, any of these tests are negative, you say it is like this particular patient is wild type for EGFR, ALK-ROS and KRAS. So the tumor board decides, you know, is it like should we stop this particular test? Then they have to test for other markers like your RET, MET, HER and BRAF. So this is how. This is a typical algorithm for lung cancer which is adapted all over. Now first we really understand, now we have really understood you need a particular sample. You have to go for molecular designaturing of the particular tumor to move further for a diagnostic, for stratification, to predict the relapse. So whatever it is. So now how do we go? So where exactly is the sample? So first exactly the preferred sample is the, first initially, traditionally, conventionally, until now it is like, for example, this is from the lymph node, definitely the tumor. So this is one, but the biopsy of this particular tumor. So where they do an FFPE. So it's a FFP is nothing but your fresh frozen paraffin embedded samples, where and it is can be stored in room temperature for long. But the problem is the patient has to be subjected to an invasive procedure here. It's not so easy, especially you want to see the for the follow-up patients. You really can't say that can we really can I go for it's not so easy like how you take a blood or a sputum or a urine sample like that. So it's not very much so easy. So this particular tumor biopsy is invasive, it takes longer biopsy. It could be because sometimes because given the heterogeneous mass of your tumor, right, as I always tell, it may not be very sensitive. So suppose the two different bits, sometimes you end up having the same from the same patient from two different bits of biopsy. One bit may show wild type. Of course it's not very, but sometimes it may show mutant. And it of course it takes a high cost of sample extractions, and then it is very well clinically validated. It is an age old golden standard conventional methods, and it's not only that, you can even go for other histopathological markers like your HER2 or different other markers, whatever you want to take it up. And of course it is not capable of assessing the tumor evolution. It can't really how the tumor, no, and it's not and you cannot really monitor no real time monitoring of the drug response. Suppose you want to check for the drug response, this particular biopsy, the conventional method, you will not be able to do it. And you cannot subject the patient to repeated surgeries, and it does not as I said, does not reveal how heterogeneous your tumor is. Whereas when you want to come to liquid biopsy, which I will really definitely go detail in all this particular session, it's like your blood. So these are all your entities which you look out for. You look out for ctDNAs, you look out for CTCs, then your EVs. I'll explain all of them what exactly are they in detail. So basically your liquid biopsy is particular this, where you just take a 5 ml or a 7 ml required to extract or enrich this particular entities from the blood, and from this particular entities you can really go for your qPCR or your digital PCR, ddPCR, and then you can go for the comprehensive sequencing analysis. So basically, so the patient is coming back to your clinic after 2 months of his chemotherapy, and you want to check is there a chance for this particular patient will have a relapse, or how does it go. So this is a very easy. You just need 5 to 7 ml of this blood. So this liquid, so that is why we call it liquid biopsy, where it is minimally invasive, it takes less time, it is highly sensitive, because you look for this particular analytes and the biomarkers which are very much specific to that particular cancer type. And it is low cost of sample isolations, but not always, and it does not provide and the biggest advantage is you will not have your pathological evaluation markers, but it can it really monitors. You will be able to monitor how the tumor evolves, and it is a real time monitoring for all your drug response. For example, you give a patient one particular drug and you want to check is he responding, so you can have definitely you can use this particular entities as markers. And then it requires the spatial, and it definitely reveals here lot of spatial and temporal tumor heterogeneity. So therefore liquid biopsy is a simple and non-invasive alternative to surgical biologies, which enables doctors to discover a range of information. You just not have only a simple positive negative. You can have a lot of information from a simple 5 ml taking a blood sample. So this is like because you have traces of your cancer DNA in the blood sample. So therefore research on this liquid biopsy SS is really gaining lot of momentum, and it has expanded rapidly, and many different biomarkers in distinct body fluids, not necessarily your blood, you can even have your saliva, urine, all that so it can be they can be, especially this is very relevant in your solid tumor panels. So because of this repeated whenever the patient comes for chemotherapy, say comes once in 2 months or twice in a month he comes over. So you can take blood samples and you can monitor the treatment response or disease progression. So what are the different entities we analyze in liquid biopsies and their application? So you can have various analytes analyzed in liquid biopsies. For example, your CTCs, then your ctDNA, your exosomes, and then your tumor proteins. So now for decision for a particular template, we usually rely on this analysis of the tumor tissues for the expression of your particular or a particular gene or a specific genomic aberration or alteration. So usually these are mostly analyzed by IHCs or FISH, that is fluorescence in situ hybridization. So this is like this analysis is usually restricted to this particular tumor alone. But however, sometimes this tumor may not be really accessible. So this, and I mean accessible in the sense for a biopsy, and these tumors, as I said, they are always changing, heterogeneous over the time. As so as the course of the therapy, the tumor evolves. Either it may really become more resistant to, or the patient may respond to that particular therapy. So this thus the metastasis at the time of relapse. So they may have suppose if at the time of relapse there is metastasis, it will have a different molecular characteristics than the tumor of the primary origin. So this thus restaging of metastasis by biopsy is possible, but it is invasive. And some size such as your bone or especially the brain, the gliomas, and for them to do a resurgery, to do a rebiopsy is a little bit difficult. Thus, thus there is like a so you have found a solution now, a liquid biopsy, to analyze this particular entities. So what is it? What do these entities provide? So you can really configure if there is a therapeutic resistant, as I mentioned. So you have the mRNAs, then you have to look for the non-coding RNAs, ncRNAs, then your RNA splice variants, and then you look for the mutational profiles, which is very important, and then you look for molecular diagnosis, and then as I mentioned, your personalized treatment strategy from this particular analyze you can really adapt, and then you can even look for the different all this variations, whatever we looked into the basics of cancer. So then you can even look for your disrupted DNA, like your and your epigenetic changes, then your translocations, your CNVs, very importantly the copy number variations, especially for leukemias. You can really look into this, and then you can even ascertain your tumor heterogeneity, and then as recollect our first two slides, we can stratify them or you can classify the tumor subtypes. So the first class what we are going to look into it is your CTCs. So this is your circulating tumor cells. So from how do they originate? From the primary tumor, but they are distinct from the primary tumor cells, with they have the EMT transition properties. Recollect our first sessions, basic sessions, where we said about epithelial mesenchymal transition. So that is why because of this transition only, these tumor cells, they are able to break away from the primary tumor and then they are able to facilitate intravasation. That is how they are able to enter the main blood stream, and then they are disseminated in the clusters of circulating tumor cells. So just keep in mind all throughout this session, your CTCs is nothing but circulating tumor cells, CTCs. Sometimes these CTCs, they may perish in the circulation. It's not like that once a cancer cell, it can survive anywhere. Sometimes so they can even perish in the bloodstream itself, and then maybe they can infiltrate distant organs. So this is like this interaction between the CTC and the blood environment is very critical. How the CTCs escape the in the immune surveillance in the blood. So this is like, as such, this particular property is very well associated with that metastatic mechanism of the CTCs and the metastatic property of that particular tumor. So it took a long while for researchers to to identify how this CTCs, the critical role of CTCs in this particular cancer metastasis. So because why is it took a long while? Because it's a very technically difficult to extract your circulating tumor cells, CTCs. So this is exactly your CTC. Then you even have this is all your healthy cells. Then the phagocytosis. Then this is your mutant DNA. So along with the other cells, the CTCs are also present. So you really need to have a technique which will enrich only your circulating tumor cells. So this is again like how this particular diagram says how the release of single CTC or CTC cluster from the parent tumor, it will allow into this invasion into this particular cluster. So they will survive. First thing, as I mentioned before, they have to survive in the circulation, and then again you have a fraction of this disseminate where they have to establish extravasation at the secondary site, where the fraction of this disseminated cells can enter dormancy in a distant organ such as your bone marrow, and then they regain again the tumor properties after particular time, and then they will be like establishing metastasis over here. So this is the particularly of the importance of how exactly your circulating tumor cells are present in the blood. So basically, if you have a very high end MRIs or your PET CTs, usually you will be can detect your primary tumors and metastasis that are very well established. So that imaging. So to be visible in that well in your PET or your MRI or PET CT or your MRI, the cell should be greater than 10 to the power 9 number. So just imagine, by the time. So but whereas using your liquid biopsy or the CTC technology, suppose you are able to enrich this particular CTCs, isn't it fantastic that in the DNA level itself, even when the cell quantity is very low also, you will be able to pinpoint it as a early diagnostic tool. So this again, it's just a simple how exactly what I have mentioned in this, so basically that is the so usually the complex metastatic process, as we have always discussed right from the beginning, it includes tumor cell invasion in your primary site, and then extravasation into your intravasation into the circulation, then survival in the circulation as your CTCs. So this and then so why I am talking so much in detail about is like so they usually are assumed to be the substrate of metastasis. So for example, a patient is undergoing a therapy. So can we not look for this CTCs and then look for like, is there a possible cause of this particular is the patient? It can be used as a scale to evaluate the response of the therapy. So this is like so because of this as I mentioned of this technological challenge of enriching them. So this CTCs have been a little bit tough, but still the recent technologies with many other tools, they have been able to enrich CTCs. So in this slide we will see how CTCs are enriched. So because you have other cells also, so blood, you have other cells in the blood. So now what are the different methods, so based on their physical properties, CTCs they can be separated from the other cells like your size, density. So and then sometimes the CTCs have the specific antibodies, and then that is how there is like they can definitely bind to this particular antibodies, and through the immunoaffinity you can clearly say that yes, you can enrich CTCs. And very fantastic tool microfluidics, which is really emerging now, you know, where it's like, you know, where it can assist, you can really use this to filter out your CTCs from the rest of the other samples, like from your other blood, like your plasma and your this one serum. So, detection and characterization of the CTC involves various techniques that require, that employ primers. So, this is a sequence dependent. So, what is do you do further after enriching them? You can go for the sequence dependent techniques, or maybe like either or maybe a deep sequencing. So, that is like you go for the digital PCR, then you go for pair, that is your personalized analysis of your rearranged ends, then the TAM sequence, that is your tabbed amplicon deep sequencing, then you go for your cancer personalized profiling by deep sequencing. So, and then your safe sequencing system, like your beaming. So, your based on all this, you know, you can draw clinically relevant information which can aid during the progress of therapy, or it can aid in therapy for regarding this particular tumor. So, then you can even also go for whole genome sequencing for the CTCs on the whole exome sequencing. So, as I said, it can help in all this. This particular utility is for diagnostic, prognosis, then monitoring, therapeutic, and then one very important thing is, can you look for therapeutic resistance. So, this is how that is like, there are three subsets, you know, that is like usually your epithelial in the transition. This is all the different subsets of your CTCs. One very important technology which is employed is your cell search system, or your CS, which enables your enrichment and enumeration of your CTCs, circulating tumor cells. So, now, like, you know, so this is like we know that it is well established that association of CTCs with the poor clinical outcome for cancer patients, it has been demonstrated in several clinical studies. So, for this identification of these targets, and then stratification, you know, and to know underlying mechanism of resistance, you know. So, it's so this is very important to analyze the primary tumor. However, sometimes, you know, for this especially for this particular primary tumors, like your sometimes even the brain, you know, the lung, you know, so that is why there is so we are using this particular cell search system. And then there is a lot of this for clinical applications, you know. So, basically this particular system, that CS system, it provides it's like it provides an opportunity to apply an assortment of accompanying tests, you know, and techniques for further characterization of and classification of your CTCs, and then enrichment of this and co-enrichment. It usually co-enriches the other leukocytes. But however, many of these particular tests have been identified in larger populations also. So, you have the well designed, so from this so you have well designed instruments for isolation of single CTCs from the using the cell search system. And so that is like, and then you even have CTCs enriched by your profile kit, and they allowed they allow a lot of your comprehensive analysis of this individual tumor cells. See, isn't it beautiful? Like, you know, when you go for a biopsy, you have a lot of tissue, a tumor tissue, which may be so many cells. It may even have your other tumor environment cells, like your epithelial cells, your fibroblasts and all that. Whereas when you have a technology, when you can really take a homogeneous cells, you know, here, so there definitely to search for a very good molecular marker is it's quite easy. So that is why the whole diagnostics of cancer is moving towards this particular directions of liquid biopsies. So that is why, you know, so usually from so CTC is not limited to merely detection of, you know, usually the cell search system is not only for the detection of CTCs from cancer patients, but also you have to use an appropriate kit in that particular downstream process, which will absolutely aid in the diagnosis or molecular characterization of your CTCs. So here you have the even your CTC CXC kit, you know, where the deep array and micro manipulator for the and the Morpho cell sorter, cell erector. So you can even go for your FISH here, you can go for your mutational analysis as I mentioned before. So usually sometimes it is for your low expressing antigens, all this particular mutational analysis can be used. So now slowly we will move to another entry which is called cell-free DNA or circulating tumor DNA. For precision oncology, it is very important to know what exactly a cell-free DNA and a circulating tumor DNA is. So what is cell-free DNA? It is usually comprised of your double-stranded DNA, which range from 150 to 200 base pairs, and it circulates mostly in blood. So its release usually happens via the apoptosis, necrosis, and on phagocytosis, and so that is how it releases happens. So in normal healthy individuals, the cell-free DNA usually originates from the usually hematopoietic cells such as your WBCs, erythrocytes, and your endothelial cells. So normal tissue sometimes which undergoes damage like your trauma or infection or ischemia or inflammation, that time also cell-free DNA could be present. So active secretions like the via the exosomes or protein complexes will contribute to the cell-free DNA. The thing with this, the important disadvantage with this is it has a very short half-life ranging between 16 minutes and maximum till 2.5 hours to maximum till 200 minutes. But and then it gets it is cleared from the circulation via the nucleus action, and it's excreted into the urine. So suppose the same, imagine a patient with cancer going for liquid biopsy. You extract the cell-free DNA from the plasma, which is now very simple, and then you go for analyzing your first thing, how do you analyze your cell-free DNA and the ctDNA, one more term, and then quantifying your total cell-free DNA, and then analyze the mutations in your cell-free DNA. For example, all the lung for the treatment therapies, they are using the T70, 790 mutations to check. So this is like during the course of therapy. So again, very importantly in precision oncology, the importance of liquid biopsies is the need of hour now. And then again you can even go for analyzing and quantifying the methylation patterns of your ctDNA. So the clinical this particular for this particular cell-free DNA includes your screening or diagnosis, then to stage molecular signatures or to select for treatment selections, then treatment monitoring as I said, and very importantly your treatment resistance and early recurrence or detection. So this is like now whatever we call this, this is now like all these markers, they have been recommended as companion diagnostics test. CDX is a very important terminology for precision oncology by this European medicine agencies and your FDA. So this is like all this particular molecular signaturing, it's like very important to guide the therapeutic decision making. So you have the fraction of that cell-free DNA. So that particular fraction of that cell-free DNA that is derived from the tumors, it is named as your ctDNA or your circulating tumor DNA. So we are introduced to three different concepts now as of now: CTC, circulating tumor cells, then you are coming up to your cell-free DNA, and then your fantastic term ctDNA. So this has that is like now over several course of the time, you know, if you see that the cancer patients and the healthy individuals. So that is if you see the length distribution. So cancer patients represent a more fragmented pattern and consequently shorter pattern than your healthy patients. So this is like, this is your first point, a tick mark, you know, to leverage to detect cancer without previous knowledge of your genomic alterations. And so here this itself you can monitor, this can be a primary marker to monitor the evolving of the tumor. So cancer patients have high level of your cell-free DNA in their serum or plasma because of more number of cells first thing, and more number of cell necrosis or apoptosis. And first very important factor is tumor cells tend to divide more faster than your normal cells, right? So that is why cell-free DNA is released in high proportion. So usually in the last few years, you know, cell-free DNA and your ctDNA have gained lot of attention as novel blood markers, and as a quantification and kinetic analysis of cell-free DNA and molecular profiling of this particular ctDNA has been suggested for both predictive and prognostic values for many of these cancer types. So it's just not like, you know, so you have your plasma DNA, then you have your differentially phased ctDNA fragment ends. Then you can even clearly go for cancer. You can clearly identify your cancer driver genes expression using this particular ctDNA, because it's the DNA which is originating from your tumor. So just isn't it like a very high and easy game to imagine itself, isn't it, like where you can match your genomic coordinates right only from the blood samples, isn't it? So you have you can clearly say what is the least expressed genes, then what is the highly expressed genes here. So just a continuation of my earlier slide, what is the source and metabolism of your cell-free DNA, where it's like, you know, so it is found in body fluids such as your blood, and it is derived, it is coming from the three main processes from both from normal cells such as your WBC as well as your tumor cells. So where there is apoptosis, then you have the necrosis. So that is, which is like active your caspase dependent apoptosis, and then so your necrosis will result in larger fragments of degraded DNA, and this process will require phagocytosis. So that is your digestion of this dead cells. So there is also an process of active secretion that releases cell-free DNA into the circulation within either vesicles, such as like either it is through your exosomes or bound to your protein complexes. So as mentioned, this half-life is very low, 16 minutes or only 2.5 hours. And then it is eliminated from circulation by your nuclease activity or by uptake in the liver, and then degradation by macrophages, and then by slowly excreted via the urine. Now this is just, you know, very first history, for example, how, like, you know, how the first detection of the tumor derived DNA or your ctDNA is first coming back from way back in 1948 by this by Sorensen. So usually so different over the years, you know, cell-free DNA in different cancers has evolved with the effort of several researchers, and then they have able to detect mutant allele frequencies up to 0.01%, And then so for it's all like small steps, where now we are able to use ctDNA for as a very important diagnostic, especially with lung cancer, to monitor your T790. So once patients during the course of therapy for lung cancer, especially if they are with an EGFR mutation and they are on a TKI inhibitor, so during the course of this inhibition they develop the resistance. This patients don't respond to this treatment. So then, do you have any other mutations like your T790? So this T791 has been 90 mutation can be checked using your cell-free DNA in the from the extracted from the plasma, and this is very well approved by the FDAs. So this is like whatever the gene expressions and all that, so several groups have been in part. So now what are the main samples? So how do you get your samples? So now what are the main steps for blood sample processing and cell-free DNA extraction? So first, as I mentioned, you need EDTA tubes. So or any of this stabilizing tube, so which are company specific, and then you can store this blood for up to 4 hours in your room temperature, and this particular tubes, they can be stabilizing tubes for up to 14 days, which are specific for your ctDNA. And then centrifugation, like sometimes 12,000 G for 10 minutes or for 16,000 G for 10 minutes, 2 steps of centrifugation you do, and then you aliquot it and then store the plasma. So this is all steps where you will have flexibility with your liquid biopsies. And then so now so first one important thing is that your blood collection tubes is very, you have different types from different manufacturers, such as your Streck tubes from the BCT and the cell-free DNA collection tubes from the QIAGENs, that is your Paxgen blood. They are called the CF DNA tubes and the Norgine tubes. So they have these companies have their pre-coated with the preservatives, So all this particular tubes they are having with their, of course, pre-coated with the preservatives as well as the anticoagulants. And then they have a typical recommendations how exactly you have to do the centrifugation. And as I mentioned before, the storage time also can be varied according to this particular manufacturing. So then once you separate the plasma and the buffy coat, so then will be usually it's a 2-step procedure, and then after that it is usually the extraction of this cell-free DNA usually happens via the silica based membrane columns magnetic beads. So this is nothing but in all your QIAGEN, for if you are familiar with your blood extraction, DNA extraction from blood using your blood mini kits and any which are mostly column based, which are silica column based, they have a high affinity columns which can bind to the DNA. And then they even have the magnetic beads which can really bind to your DNA. So then you look for so sometimes this is like even you even have automated systems also, for example like your Promega. For example, you just put your blood sample into the kit and then the whole they have a wonderful machines or platforms to extract your cell-free DNA and to give your best quality and quantity of this particular ctDNA. So based on this extraction kits also, you have the different yields also vary. So according to the custom lab, you can really choose based on your economics, the cost also, you can choose what exactly kits you are going to employ. For example, your Promega or the AirPlat Biomist systems, that is which has your MagMax cell-free DNA kits. So they as I mentioned, these kits will be working with different columns. So after you have the quality control where you quantify and then you qualify and then go for this analysis. the downstream analysis. So this is like from the colorectal cancer, for how it is really very well relevant in the clinical decision making, you know. For example, colorectal cancer releases your DNA fragments into the circulation called cell-free DNA, and this cell-free DNA mixes with ctDNA, and then it mixes with your physiological cell-free DNA. So physiological cell-free DNA, so this is your ctDNA, and then after this is detected via the detectable in the peripheral blood sample. From the blood you can take the ctDNA, and then you can assess the tumor month and over the time. So you can clearly see how and during the treatment response also. So at the time of surgery and during the time of at the point how he has at the point of initial before the response, that is at the 0 stage, and then at relapse. See how you can clearly see how the ctDNA levels clearly increase as the time progress. So this is a very good marker for to monitor your colon cancer disease stage. So this is like different methods, like with other also, like with the CTC also, as I mentioned you have. So for the ctDNA, there are many commercialized methods are available for monitoring the genetic alterations in this particular ctDNA. So for the point mutations, then the copy number variations, your chromosomal aberrations. So for the, you can do the PCR, you can do the RT-PCR, digital PCR, SNP arrays, and then you even have the second generation sequencing for your ctDNA. And then you even have your multiplex ligation dependent probe applications, then your molecular combing, then you have the comparative genome hybridization to identify your CNVs, copy number variation, and chromosomal aberrations. So usually this cell-free DNA, several studies, you know, they have highlighted the importance to perform. Always it's important to check your QC, quality controls, on the isolated circular DNA. Suppose it's like and if it's suppose if it's highly fragmented, and it should be, its size should be ranging from 22 and 220 base pairs with a minimum peak at 167 base pairs. So that is like it is like it corresponds to the length of the DNA which is wrapped around a single nucleosomes. So that is like you have to quantify and qualify this particular cell-free DNA or the ctDNA, and it should be you should not be able to confuse it with your genomic DNA. So this is very, very important. So capillary electrophoresis was used to measure the size of your DNA fragments before, and then it is able to give your estimate of absolute concentration of the ctDNA or the cell-free DNA. And then, as I said, you know, you have the qPCR and the ddPCR methods, which can evaluate the amplificability of the cell-free DNA, as well as your concentrations and the integrity, which is very, very important. So that is so this is like it should so for example so there is absolutely, there are several assays are coming up in the market which may not really require a reference sample and a calibration curves. So but very, very important to keep in mind your copy number alterations, as well as your genomic DNA contamination into mind while going with the ctDNA. Now, very interesting, what exactly after, now you have come up with a liquid biopsy, take a blood, that is also very fantastic. And then after you get such loads of DNA, the first thing what a molecular biologist now would really go is for next generation sequencing. So this is your backbone, this is the very fundamental aim or the fundamental endpoint need for the ctDNA, for be it for lung or for breast or for cervix or anything. So basically this is like suppose you start with your whole blood collection in specialized in your CS cell-free DNA tubes. The plasma, you separate the plasma which contains your cell-free DNA via centrifuge, and then you extract the cell-free DNA from the plasma. So usually then you go for simplified Illumina library. So this all we will be learning in the further sessions in the future session, exactly of what is the library preparation and all that for NGS, and then Illumina library fragments as a result of your NGS library preparation. So here the dark and the green blue plus represent the Illumina adapters here for the P5 and P7, which will really hybridize to the flow cell and subsequent bridge amplification after ligation to the cell-free DNA fragment. So samples specific indices which are used to identify the patient samples are typical in your this one. So they are particularly typical in your in the dual format, and here they are shown as your I5 and your I7. So you have the again your unique molecular identifiers, that is your UMI. They will serve as your molecular specific barcodes. So which will enable the bioinformatic filtration of amplification, or maybe sometimes due to the sequencing error to analyze a very high quality variant, to analyze a very high. This is a very important point. So coming to see, here you have the sequencing by synthesis SBS on an Illumina element, which allows for one fluorescently tagged nucleotide to be added to the growing read per cycle. For your G, your A's, C's and D's are tagged with your pink, pink, blue, green and yellow fluorochromes. So thus after this instrument has converted the captured images to base calls, the data is converted into your what is you call this your FASTQ files containing your reads and the quality scores. This is very important. So since the reads and the FASTQLs do not describe genomic, they may not really describe the genomic location of the read, so this particular reads have to be aligned to your reference genome. For example, here in this case is the human genome. So this is this alignment is referred to as your SAM file, which is a binary counterpart called, which has a binary counterpart called your BAM file. So the BAM file contains all informations in original FASTQ file along with the mapping information of the read, such as your genomic coordinations to which it is aligned. The BAM alignment file will also serve as the core data for diverse downstream analysis. Example, your calling of your SENAs or variance, estimation of your tumor fraction from your plasma, and then calculation of the fragment size distributions or nucleosome mappings. And this is E is your example of a clinical report summarizing the interpretation of your genomic alterations detected from your cell-free DNA. So this is very important here. For example, you have this particular genes here, where exactly the region, then which segment, then what size. So is it like amplified? Yes. And then clinical relevance. So this is how the particular report is presented, and this reports have to be so this report describes the detailed genomic alterations alongside the your allele frequencies VAFs and pathogenicity with potential clinical implications. So this should be, this particular report is like so for example, a lung cancer patient has come to the clinic. You go through this take out the, instead of going to the biopsy, you take the DNA and then go to the ctDNA and then go for NGS analysis. You get a report like this. So how will you define the outcome of the disease, or how will you do you want to have to change the therapeutic response? So this is what is being discussed in your tumor boards. and then it has to be related to the patient's clinical status or your CPS. So this is like a simple table which I have made, like for in different cancers, what are the different markers they have, and the technical methods, like, and then what are the samples they are using. So for example, in the hematological sample malignancies like your leukemias, you can take the plasma, serum, CSFs, and then is used for diagnosis, and then looking for this particular different mutations, and you can even use it via your RT-PCR real time or the sequencing, and then or your droplet digital PCR. And then for the thyroid cancer, you have the cell-free DNA quantity and integrity as your index, which can be determined by your qPCRs. And then for the colorectal cancer, you have this particular different markers. And then for this HPV also, for using the duplex digital droplet PCR, you can take the look out for the HPV cell-free DNA, and then it can be used taken from the serum. It can be, and this particular marker is used for diagnostics or predicting response to therapy. And then EBR via EBV for DNA from the nasopharyngeal carcinoma. Like this, as I mentioned, this is a fantastic example, the lung, you can even look for all this particular mutations, but for of importance is here T791, which is the most widely used in marker for the for the treatment response. Now, very important, this is all the different organizations, which is very important. Only after their approvals, any of, suppose I come up with a different biomarker, I just cannot directly use it on the patient. So I have to get approval of the different pathological associations, different oncology associations, and all this, for NCCN for your breast cancers, and NCCN for your so they have different set of different set of guidelines. For example, for any particular for plasma over serum, it is approved by the IASLC as well as your ASCO. Prioritize histological diagnostics, yes. For some, definitely they are only the recommended. Suppose you have, if the tissue is suppose the patient is in a very unfit condition to go for a biopsy, yes, that time they are recommended, these are the guidelines recommended to go for, especially in the lung, definitely to go for a liquid biopsy. Plasma genotypic at progression to detect target alteration, yes, all these organizations, they are recommending it. If plasma is negative, tissue biopsy, if the particular biomarker is, again, suppose in the cell-free liquid biopsy, if it is negative, then should I go for the tissue biopsy? Yes, it is very much recommended. All organizations recommend that. Suppose a positive actionable cell-free DNA alteration is if suppose I detect any mutation alterations in the liquid biopsy, can we go ahead with the initiative? Yes, all. So that is like your liquid biopsy has gained a lot of its own place, as much equivalent to the tissue biopsy. So is and so they recommend NGS is preferred for detecting fusions, and your report should include what is the platforms they have used and what are the difference. So what exactly the report which I have shown. Now coming to another last analyte which we will be discussing in this session is your circulating tumor RNA and the exosomes. So what are circulating? MicroRNAs, circulating miRNAs, what we call it. So they are also considered important promising biomarkers for many human diseases, especially for cancer. They meet several criteria for being a preferable biomarkers for high specific, because they are very specific, they are easily accessible, and they are sensitive. And they have been these miRNAs are found in many biological fluids, and they are stable to some extent, and they can be extracted from the blood and from other liquid biopsies, like for example the saliva for the oral cancers, and then They are also highly specific for tissue or cell types. For example, your miRNA 122.5p is highly enriched in liver cancer, and it is selective. So this is like so because of this specificity, there we are able to have a possibility of using specific miRNAs to determine the initiation and progression of the disease. So this is like, I don't want to go into real details. So basically they are nothing but your small non-coding RNAs that suppress gene expression both by inhibiting protein translation and promoting mRNA cleavage. So just very quickly how this biogenesis of mRNA and this cellular export is happening is, they are transcribed mainly by the RNA polymerase. So and for after capping, splicing and pollinated, they result in the formation of a primary miRNA, which is with the hairpin structures. So within the nucleus, this miRNA is processed by the DROSHA, that is your RNase A and its cofactor DGCR8 into 7 to 100 nucleotide pre-mRNAs, which is exported to the transported to the cytoplasm by your exportin 5 through the nuclear pores. So afterwards, this dicer, another RNase cuts pre-mRNA to a double stranded RNA in the cytoplasm, and then embraces the mRNA strands and its complementary sequence. Helicase, here. Helicase continuously unwinds this duplex mRNA into short RNA strand called mature mRNA, that is which is incorporated into your RISC complex containing an arginate protein, that is your AGO2. This incorporation will lead into the leads the RIC complex to the 3' UTR of the target mRNA, resulting in mRNA cleavage or inhibition. So mRNAs are also processed from the introns of protein coding genes by the pre-mRNA splicing machineries, that is your DROSHA or DGRC independent pathways here. So the expression of these mRNAs, which is also called MIR-trons, is correlated with the host gene expression based on their location in either the introns or the splice site junctions. So this is, they are very important in for identification of several cancer types. So this is very brief. So this is the summary of your modulated circulating mRNAs in several prominent cancer types. So you have in the gastric cancer, you have the MIR20A 5P and MIR22 were found to be increased in both plasma and serum samples. So that is like, so before the clinical 5 years before the clinical symptoms have appeared, the changes in MIR 222, MIR 378C 5P and MIR 754 5P in the serum, there has been changes, there has been increase in their levels in the serum. So that is 5 years before the clinical symptoms appear only, this particular miRNAs are increased. So now we can really understand, how this modulation of the circulating miRNAs in this several prominent cancer types is an important, it can be used as a good biomarker for during the aid and therapy during the treatment progress. So one more very important markers in from the blood, again one more are your exosomes, which are with diameter of 40 to 160 nanometers and they are lipid bilayer membrane vesicles, and which are actively released by most cells and stable in then they say stably circulate in most body fluids. And usually these exosomes are heterogeneous group of membrane structures vesicles actively released by cells. So your MBBs or your membrane structured vesicle bodies, you know, they are usually endocytic structures formed by the inward bunding of your endosomal membranes. So, just endosomal membranes. So, vesicles accumulating inside of this membrane vesicular buds, namely your intraluminal vesicles, are released as exosomes by the fusion of this MBBs with your plasma membrane. So, usually they are the new targets. These exosomes are the new targets for liquid biopsies, and they are compared to the CTCs and ctDNAs. Exosomes are like they are like bison themselves a total living cell secreted vesicles. They by themselves they are organelles, and they are more stable during circulation. So, they have been many technologies have been used to separate exosomes from various body fluids and to detect exosomal cargoes. So, several so this is very important. So now after after they are detected again by the other NGS, you even have your qPCR, RT-PCR, ELISA, digital PCR, Western blotting. Again you have Western microfluid dips which can detect your exosomes. So, how do you detect exosomes? By the fluorescence detection, then the calorimetric detection, then you have the droplet digital PCR detections, then the single EV detections, then you have the SPR detection and the electrochemical detections. You know, these are all like for any other particular calorimetric detections, they can be employed. So, they are sometimes, you know, molecular beacon, you know, so this is also very this was like they have this is very important tool to detect your exosomes. So, so it is very important to detect highly sensitive and early detection of your cancer specific exosomes. So, coming towards end of this talk, we have really seen in this whole cancer detection methods, we have slowly come to understand what exactly are biomarkers, what exactly are the different, why do you need a screening, what is the objective of screening, and then we have totally gone on to talk about different cancer detection methods for each cancer type, and then we are coming to the liquid biopsy, which is a new game changer and which is the need of the hour and which is the new changing tool for cancer detection. So, we have really come into now after at the end of this session, you will have an understanding what exactly are the entities you employ to go for liquid biopsies, like your circulating tumor cells, your ctDNAs, and then your exosomes, and what are the high end technologies which we are using to adapt for to give make a clinician make a right decision to help in the towards the treatment to give a better treatment outcome for the patient. So, definitely to improve the survival rate of the patients. So, we will really conclude by saying that liquid biopsies are the game changers for cancer testing. Thank you.